Hey y'all, my name is Prashant Kumar, or PK, and I'm an MCAT tutor at Shemmassian Academic Consulting. Today, we're going to be doing a passage walkthrough in the MCAT Biochem section. You should be taking roughly 6.5 minutes on this passage, but today we're going to go more in depth so you can see exactly what's going on in my head as I take on this section. Let's get it. Alright, great. So I've gone ahead and put the passage that we're going through on the left and the questions on the right. So feel free to take a moment and pause the video and come back to us. If not, let's get right into talking strategy. For me, I like to maintain the exact same strategy going into any passage I come across. Regardless of whether it's super easy or super hard, it gives me something to come back to and boost my confidence with because I know exactly how I'm going to tackle that passage. For me, that strategy looks like read a paragraph, write a quick 3 to 5 word summary, then go on to the next paragraph. Go on, do the same thing for all the paragraphs until I reach the end. The reason I like to do this is it provides accountability. It makes sure I understand exactly what's going on in the paragraph before I go on to the next, and prevents sort of reading random words that don't really make sense in the context of the whole passage. And I found this saves a tremendous amount of time. Sort of making sure if I don't understand it on that first read, that one paragraph, I can reread the paragraph as opposed to the entire passage. So that's my strategy. Additionally, I don't like to spend too much time on figures. I like to give it a quick gloss, but nothing too in-depth. The reason for this is I want to make sure that I spend time where I need to. If the questions don't ask about the figures or have no need for me to look there, I'm not going to need to look at them. And if I do need to look at the figures, it's inevitable that I'm going to have to come back to them. So I don't want to spend too much time on that first read. Maybe get a quick glance at what's going on, maybe the axes if it's an experiment, sort of what the trends are, but not going too much into depth. All right, with that, let's get right into talking about the passage. In contrast to traditional thought that the purpose of mRNA outside the nucleus served coding functions only, scientists have begun to appreciate a new class of long non-coding RNAs, LNC RNAs, that are stable mRNA gene products. The expression levels of LNC RNAs were determined in an NCI-60 cancer line using a novel assay. Specifically, LNC RNAs, ANRIL and PVT1, were studied. In NCI-60 cells, ANRIL has a copy number deletion, while PVT1 contains a copy number amplification. PVT1 contains a heavily folded and polymerase inaccessible N-terminus. The relative expression of these transcripts was determined using RT-qPCR as shown in figure 1. And then it gives us a figure right there. But whenever I read a paragraph, my first question is, what is happening? It's a pretty broad question, but I want you in the simplest words possible to explain exactly what's going on in that paragraph. So for me, I'm here looking at LNC RNAs, right? This new type of RNA that they're looking at. And then we have ANRIL, I'm just going to call it A. And then we have PVT, I'll just call it PV. And so what's going on? Why are these two special? Well, this is an X, that's a deletion, and this one's an amplification. This is a plus. And so with that, the question arises is, why are these important, right? What is happening? Well, these are looking at what is going on with sort of the expression of these cancer cells and NC60 cells. And so that's all I'm writing. Is like in NCI-60 cells, what is happening? What is the expression of these transcripts? All of that. And so this is the exact sort of diagnostic technique I'm going to use for this large paragraph. There's a lot of details. We're probably not going to get all of those details on the first read. But what we can do is understand, here's the paragraph where they give us the introduction of ANRIL and PVT1. Nothing beyond that. We understand LNC RNAs are these different type of RNAs. None of that. These are all details that we'll come back to if needed. All right, and so next we have our little figure. Again, I'm not trying to spend too much time on a figure. All I see is, okay, we have these cell lines that are coming from different organs in different parts of the body. We do a reverse transcriptase, qPCR, so some sort of quantification of these transcripts. And then we have expression profiles. Remember, I want you to familiarize yourselves with these experimental techniques. Understand exactly what they're measuring, how they're coming across these measurements, because while it might not be important for this passage individually, understanding them, especially on a free response question, could be the reason you get a question right. So right now I'm not going to spend too much time on understanding RT-qPCR or what these expression profiles are, but we just understand that this is some sort of quantification technique. All right, PVT1 overexpression is associated with several types of cancers. The gene is regulated by p53 through a canonical p53 binding site. The LNC RNA is likely involved in the upregulation of proto-oncogene MYC. The relative expression levels of ANRIL and PVT1 were plotted against normal gene dosages as shown in figure 2. All right, so again, big picture understanding what is going on. And so the idea is that PVT overexpression is associated with cancer. So PVT plus, and then you have cancers, right? And so how is that occurring? Through MYC. So I'm going to write MYC increase, right? This proto-oncogene is upregulated, and that's really all I'm focused on. PVT1 amplification results in cancers with an increase of this MYC proto-oncogene. And then we see these expression levels with these graphs. And again, I'm not going to spend too much time. Maybe look at the x-axis. Okay, we have ANRIL and PVT, deletion and amplification, sort of the defining factors, and then their expression. All right, we'll come back to it if needed. We're not going to focus too much on it right now. After verifying the effect of copy number on relative expression, researchers decided to target PVT1 with a novel therapeutic in the cancer cell line. PVT1 forms several helical structures as shown by circular dichroism spectroscopy. Scientists developed an inhibitor that breaks several important helical interactions. To characterize the effect of the inhibitor on protein folding, researchers added the inhibitor and carried out circular dichroism spectroscopy. All right, so exactly what's going on, right? Complicated words. When they said words like dichroism, I don't know what that means, right? This hasn't been in our content studying, and a lot of people are going to get scared. Don't be scared. PK says you're not allowed to be scared, so you're not allowed to be scared. But PVT1 is what we're looking at, right? That's a common term that we've seen over and over again. And so what is our basic idea? Well, we have a new therapeutic, right? Therapeutic, we're going to inhibit PVT1 via helices, okay? Right? So the therapeutic is inhibiting PVT1 because of helical interactions, and then they're doing spectroscopy for sort of quantifying that. But I don't want you to get bogged down in the details, because the details can be scary. Big picture understanding, take a deep breath, and we're on to the next paragraph. Another researcher hypothesized that ANRIL underexpression may lead to tumorigenesis. The researcher delivered exogenous ANRIL LNC RNA to NCI60 tumor cells and measured cell viability. After several weeks, there appeared to be no effect on tumor growth. The researcher then switched to non-cancerous cell lines, and after conditionally knocking in tumor drivers in both cell lines to induce tumorigenesis, the researcher found that cells with a normal dosage of ANRIL survived longer. All right, so what is happening? So we have experimental setup, right? And in a big picture understanding, they're looking at ANRIL in two cases. One, NCI60 cells. Two, normal cells with knockout genes. And then we see some sort of positive improvement here. And then here we see nothing, right? Net neutral. There's no effect on tumor growth. And so these are the types of breakdowns I want you to do. I want you to have these complicated paragraphs in simple words, right? I don't care if you use abbreviations. I don't care if you use arrows or diagrams. Whatever is easiest to help you learn, do it. No one else is going to be reading these notes other than you. With that, let's get right into question number 1. Question number 2. Question number 1. A researcher decides to PCR amplify PVT1 using an N-terminus primer. The experiment fails because A, N-terminal protein secondary structure prevents the primer from binding. B, N-terminus primers are not functional. C, N-terminal RNA secondary structure prevents the primer from binding. Or D, more than 2 primers are needed for the experiment. All right, so what is the question asking? Well, it's asking, why is the experiment failing when we're trying to amplify using an N-terminus primer? That's the understanding. How is it failing? And so, what is special about PVT1 and N-terminus primer? We should remember that we read about N-terminus primer in a paragraph, and that was in the descriptive paragraph right at the beginning. So we're going to hunt back there, and we see that here is where it talks about N-terminus. I don't expect you to remember the detail of when they talked about the N-terminus. What I do expect you to know is that they mentioned it in the first paragraph, and we can come back to it. So PVT1 contains a heavily folded and polymerase inaccessible N-terminus. The relative expression of these handships is determined using RT-qPCR. So this is the big paragraph, big sentence. PVT contains a polymerase inaccessible N-terminus. So why would that make sense, right? Well, the experiment would fail if the polymerase cannot access it or the primer cannot bind to it. So right away, we're deciding between two answer choices, A and C. Note that B, N-terminus primers are not functional, doesn't really make sense, right? We've seen N-terminus primers work before, we just haven't seen that, and it doesn't mention that's a reason why these wouldn't work here. And in D, more than 2 primers are needed for the experiment. Nowhere does it say that. It never says anything about the number of primers needed. And so we can eliminate D. And so now we're down to N-terminal protein secondary structure or N-terminal RNA secondary structure. And this is where an understanding of the experiment, big picture, we don't even need to focus on the little picture. Big picture understanding. What is PVT1? PVT1 is a type of LNCRNA, right? It's an RNA molecule. That was like the whole big picture understanding, is that these ANVIL and PVT1 are these special long non-coding RNAs. And so without even going back, we should be able to be like, oh, that means this isn't a protein. This is an N-terminal RNA. And so with that, we've eliminated 3 answer choices pretty easily, and we've picked the reason why the experiment would fail. And with that, we're on to our question number 2. Okay. A researcher knocks out PVT1 in the NCI60 cell line. After the knockout, relative expression levels of MYC shown by RT-qPCR likely A, increase, B, decrease, C, remain the same, or D, increase only in the case of a full knockout. All right, so we talked about MYC earlier, right? We actually put it in our summary. So why don't we just go back here? Here we see in our summary, we don't even have to go back to the paragraph. We see that PVT overexpression, right? PVT plus results in increase of this MYC proto-oncogene, which results in cancer. So the main sort of understanding is write out the diagram. PVT is directly related to MYC levels, okay? And so here, the experimenter is knocking out PVT1, and then we're looking at what levels of MYC happen. What happens? So I like to think of this in terms of arrows. Keep it very simple. If we draw a downward arrow, right, and the downward arrow continues directly down, that would mean we'd have a decrease, right? Decreased PVT would result in decreased MYC, just in the same way increased PVT would result in increased PYC. Since they are directly related, the arrows just follow, right? If we have an increase here, we see this is going to also follow this way, and it'll go down here, and it'll increase that. And so here, since we're knocking it out, a knockout means we're decreasing the amount of expression, we're going to have a decrease in MYC. And so B is going to be our answer. Not A, not remaining same, and not increase at all. Like we know A and D are automatically out because this should be decreasing. And so with that, we see how we took a very complicated problem and just made it into one simple arrow, right? And if we can draw, we can identify if one side of the arrow decreases, then that means the opposite side of the arrow also decreases. All right, question number 3. All right. The information in the passage suggests which of the following about the effect of copy number on relative gene expression? A, copy number is inversely correlated to gene expression. B, copy number only has an effect on gene expression for LNCRNAs. C, copy number only has an effect on gene expression in highly regulated cells, such as NCI60 cells. D, copy number is directly proportional to gene expression. All right, copy number is something that has been mentioned over and over again, Why? Well, we should remember from our sort of summary right here that what is happening to the copy number when we're dealing with anvil and PBT1, right? We have a deletion in the case of anvil, so a decrease in copy number, and we have an amplification or in the copy in PBT1, so extra or overexpression. And so that's the idea, right? Anvil, a decrease, and PBT1 increase. That's like the big picture understanding I want you to think. Okay, so now what is happening, right? If anvil is decreased, right, we're looking at our, sorry, I've been saying anvil, it's actually anril. If anril is decreased, right, you have a deletion, what is happening to the relative expression? So we see that there is a significant thing, right? P-value less than 0.05 means significance. And so compared to the normal, there is a decrease, right? Remember, we try to keep it super simple, right? Annual is a decrease already, and that means a decrease in the number of copy numbers will result in decreased relative expression. And so right away we see, okay, decrease in copy number means decrease. Let's see what happens when we increase the copy number as related to PBT1. Well, we know PBT1 on its own is an increase. But what happens when we have that amplification? Well, we see there's this shift. It increases, right? Just as from normal to deletion we went down, from normal to amplification we go up. And we see that it's significant in the same way. P-value less than 0.05, significant. And so we see that, okay, a decrease in copy number results in a decrease of expression, just as an increase in copy number results in an increase in expression. That would mean that what? This relationship is directly related, right? If the arrows go in the same way, just like we saw in the previous example, then it's directly. Now, if the arrows are pointing the opposite way, then we could deal with inversely related. And we see that our answer choices suggest exactly that, right? D makes sense, right? Copy number is directly proportional to gene expression, not inversely correlated. B, let's look at B. Copy number only has an effect on gene expression for LNC RNAs. That is not mentioned anywhere in the passage. In fact, I doubt that is true in most cases, right? Copy number can affect anything, and we should always be hesitant about using words with only, right? Only what, right? Only is a very extreme statement, and it's likely that there's always going to be some example that runs counter to the claim, so we can eliminate B. Copy number only has an effect on gene expression in highly regulated cells, such as NCI60. We actually disproved this in this final paragraph, remember? We used normal cells here, and we saw that anvil had an effect on it. And so, again, be careful with these statements that have only in it. And so we know that C is not true. And with that, we've eliminated A, B, and C, and we're left with D, and we're moving on to our final question. 3 for 3 already, so let's crush this final question. Perfect score time. 4. Tumor cells are shown to upregulate PBT1 in hypoxic conditions. What is the most likely the terminal electron acceptor in metabolism during hypoxia? Is it A, oxygen, B, water, C, pyruvate, or D, ATP? So right away, we should realize this is a standalone question. Nothing we looked at in our passage is going to be helping, because the question is, what is the most likely the terminal electron acceptor during hypoxia? And so this is the word I want you to focus in on, hypoxia, because it's giving us a setting that we really need to hone in on. And what is that setting? A low oxygen state, right? When we don't have oxygen, what is going to be the terminal electron acceptor? And so this is under the field of metabolism, namely under glycolysis, right? And anaerobic respiration. When we don't have oxygen, we're only going to be dealing with anaerobic respiration. So these glucose molecules are not going to be able to be broken down all the way to our electron transport chain. They're not going to be able to make it to acetyl-CoA and move all the way down there. Instead, what's happening is we move all the way to pyruvate, and we're kind of stuck there, right? We move from glucose to pyruvate, and we're stuck for a second. And what happens when we're stuck? Well, the pyruvate takes these extra electrons and provides an alternate path of energy production. And so these electrons move from NADH and form onto pyruvate. And so this pyruvate is the final electron acceptor. Note, it's not going to be oxygen because oxygen doesn't exist. We're very low on oxygen. Water is not true and ATP is not true. This is a standalone fact that you need to know. All right, great work, team. We crushed another biochem passage. Kudos for you for getting it done. Super challenging stuff, dealing with experimental designs, amino acids, sort of Western blots, all of these sort of concepts that you need to know for the test because they're in that biochem section and they can come up on the biochem or even the chem phys section. If you like this video, give us a like and comment down below exactly what you want to see next. Definitely check out our 528 series playlist on the channel from Vikram. Super helpful tips and tricks and strategies to help you get your maximal score possible. And finally, click on the link in our bio to sign up for our MCAT question of the day so you don't miss out on any practice before test day. Happy studying, and let's get it.